what I'm going to talk about now is the work going on at the Ivy Center, and I'm going to also finish with the work my group has done with the CMV story, which has been totally independently done from what Cecilia's done, but we're finding things that are supporting her work and vice versa. So my first caveat is I'm going to be presenting data that was generated by colleagues of mine who did most of the work that I didn't do. And actually, we were going to have Lee Hood present some of this, but I will mention as I go along. So there are three kind of concepts that I'm going to present. I'm going to try to go over it in a gentle, superficial way so that you don't have to, you can just sit back and relax because it's the last talk of the day. So don't worry about memorizing everything I say. And the first two projects are in revision in high-profile journals, so I can't actually show some of the specific data about them, but I will show you the gist of the studies. So the first study is biomarker of tumor histology, and Ralph Puchalski, who's back here in the back, you want to raise your hand, Ralph? Ralph did a huge amount of this work, and Namita Shah also did a lot of this work. Ralph was at the Allen Brain Institute. For those of you who don't know, Paul Allen has a massive $500 million institute that has just moved down to South Lake Union. And the only project they've ever done on human disease was glioblastoma, and Ralph was the spearhead of that, funded by collaboration between Ivy and Allen. So you know, we know about this classification system. We've been talking about the four canonical pathways of GBM, tumor-initiating cells, and the mesenchymal, classical, proneuronal, and neuronal. And what the Ivy GAP project has done is was the brainchild of Greg Foltz, which was to say, you know, let's dig deeply down into this and let's do a massive analysis. So basically, it started with 41 patients, 42 tumors. How do you do this thing? Okay. But as you can see, Greg would resect a large tumor. I contributed a few tumors. And these tumors were sectioned very methodically. And then every other section had an H&E, and the other section was used for in situ hybridization or laser capture microscopy. So machine learning was used to take these tumors and analyze them for patterns that are known to be prevalent in glioblastoma, like leading edge, cellular component, or perivascular, pseudopalisading around necrosis. So each of these areas were analyzed, and then machine learning was used to put that in the computer, and then laser capture microscopy was used to pull these out, send them off for RNA-Seq. And all of these patients had extensive clinical histology, clinical and pathological analyses that are all available. It's all been uploaded onto the web as a massive data resource for people interested in doing research. But it allows, just on a practical basis, anyone to go in and pick, for instance, a gene and say, I want to understand where this gene is expressed. What they did was get 50 different genes that are involved also in cancer stem cell biology and whittled it down to a panel of 20 that were considered to be the most relevant, did in situ hybridization of those and showed where these are. And for instance, you can go through tumors and say, I want to see all the VEGF-A expression, and it'll pop it up and say this is primarily expressed in areas of pseudopalisading tumor around necrosis. So I'm going to kind of gloss over a couple things, but hit the main point, which is that this analysis, and Ralph, you can correct me if I misspeak, by going through carefully and analyzing different components of the tumor, instead of saying that these patients are mesenchymal, these patients are classical, these patients are proneuronal, it actually ends up being that the patient is basically accumulation of the most prevalent histology they have, because each histology is more specific for the gene expression pattern than the actual individual patient's tumor. And I think you can see that in this next example. which is shown here. When they analyze by gene expression patterns, what you see is these different areas of tumor pull apart. So you have areas of microvascular proliferation, you have leading edge, you have cellular tumor, and you have areas of palisading areas around necrosis. And these pull apart into different components, which essentially then redefine the tumor as aggregation of multiple different expression profiles of the histology. The other main concept that has come out of this work, and it hopefully will come out in the journal, it's under review at a revision at Science right now, will be that the concept of a cancer stem cell gene expression profile may be more relevant to the actual histology where these cancer stem cells reside in the tumor versus just all cancer stem cells are the same. And there seem to be patterns, for instance, in areas around hypoxic regions where you get one sort of flavor of cancer, glioblastoma cancer stem cell, which has a lot of markers relevant to hypoxia, as you might expect, and then other areas where it's just rapidly proliferating in cells that have other key hallmarks. So as I mentioned, I'm not going to get down into the details of this, but just sort of go over the thematic discoveries. So this is the Ivy Gap project that was the brainchild of Ralph and Greg Foltz, and has been uploaded onto the web. It's available off the web for people, and hopefully will come out as a paper to kind of redefine glioblastomas down to the more histopathological localization of cells versus just as a bulk tumor. The next project that I'd like to discuss was a project started by Dhimant Ghosh, who's in the back. Dr. Ghosh was a postdoc in Lee Hood's lab and now works in our lab. And the concept was to use, to define or to try to create a biological marker of glioblastoma so that we can follow patients better than we do now with MRI, which is really a difficult, hazy approach, especially in the reclassification era of anti-angiogenesis therapies. So Dhimant, sort of using the principles that Lee Hood mentioned this morning, he kind of came up with an algorithm to say, I want to search the database for protein or genes that encode for proteins that come from brain, and also proteins that are on the cell surface or are secreted. And I'm going to whittle those down, and then I'm going to come up with a panel that are correlated with those that are actually found based on our databases of TCGA and Rembrandt to be highly expressed in glioblastoma. And then I'm going to take those, and then I'm going to exclude those that are seen in normal brain, low-grade tumors like oligodendrogliomas, and low-grade astrocytomas. So I'll hopefully have a panel of genes that are expressed from the brain on the cell surface that are secreted and are only in glioblastoma. And once I come up with those, I'm speaking for Dhimant, once he comes up with those, he's going to use cutting-edge techniques of mass spectrometry to see which ones are actually really on glioblastomas, and then see if those are related to proteins that come out of the brain tumor, go into the cell, into the vasculature, and can be pulled out of your blood. So wouldn't it be great if we had a blood test that says your tumor is recurring and we can see these proteins going up or down and can give us a signature of the presence of glioblastoma, recurrence of glioblastoma, or response to therapy. So by doing this, Dhimant did these studies, and then he compared these potential candidates to looked at glioblastomas versus normal, and he came up with several hundred candidates. And then he did a certain type of mass spectrometry where you label cells, different glioblastoma cell lines, and cancer stem cells from glioblastoma, and normal stem cells, normal neuronal stem cells, with these biotintylated probes that go to the cell surface, and did certain mass spectrometry experiments that are quantitative to figure out which ones are actually more highly expressed on the cell surface of glioblastomas. From those, he found the list that he thought was relevant, and he looked back to the database of gene expression, and he found that some of these were actually very, very highly associated, up to 100% specific for glioblastoma. And then he compared the glioblastoma versus oligo and astrocytoma, and he excluded the ones that were seen in all these other lower grade tumors to come up with a panel of 33 that were more or less a signature of glioblastoma. Then he went back and did a type of mass spectrometry called SRM mass spectrometry that's quantitative to look at actual glioblastoma tumors to see if these proteins were expressed differentially from the glioblastomas versus the non-tumor. And he did find that they pulled apart quite elegantly and pulled apart so that you could almost define if the blood came from a glioblastoma patient or actually plasma specifically from a glioblastoma patient or normal control patient. And then he collected 20 control plasmas and glioblastoma plasmas from 4 sources around the country. And he made a training run and then a validation run to determine whether these markers, some actually go up, some actually go down, could actually define in a blinded way who has glioblastoma and who doesn't. And the data ended up showing quite elegantly if you combine 4 of these, for which we had an ELISA test. We have an ELISA test, we can do all 4 of these at the same time. If you see a pattern that emerges of all 4 proteins going in certain directions, then with very high likelihood, you can determine if a patient has, if this blood came from a glioblastoma patient or from a healthy control. And this is shown in, for this marker here that goes up in glioblastoma, that this marker is increased in glioblastoma patients. So we submitted a grant, an R01, that was reviewed, and I was educated and schooled in biomarkers, which I'm not an expert in, and we've come back and focused our plan, which is to make it more of a prognostic project. And in doing that, we started collecting preoperative and postoperative plasmas over a course of time, and we found that we've had 15 patients or so far, but we're starting to see patterns emerge for each of these different proteins that we see them go up after surgery, maybe from the resection, and then start going down. And one marker that seems to be very highly specific for glioblastoma and glioblastoma cancer stem cells, we have in 10 patients, we've seen after 10 days, in almost every case, this marker goes down, and it seems to be correlated with the degree of resection. So long story short, this is a work in progress where we're going to try to go back to those 33. We've only looked at 4 with the ELISA blood test, and reanalyze those 33, come up with a comprehensive blood test, hopefully where we can have not just a blood marker like PSA or something like that, but a panel of blood markers, some going up, some going down, and tumor, and try to follow patients prospectively over the course of their disease to see if these markers correlate with the tumor presence after the resection, recurrence, and response to therapy. I think this would be a helpful adjunct to our ability to treat these patients. The last project is, you've just heard a lot about cytomegalovirus, and it's been a long, strange trip in a way that started 2002. And I think that being humble and having a lot of humility has been the most important part of this venture for me, because we really don't know what's going on, and Cecilia has just presented information that may implicate some kind of unknown viral particle or cofactor. But I'll just tell you what we're doing independently of her that seems to be corroborating some of her efforts. And the reason I think this, so it started in 2002 when we found on immunostaining that glioblastomas had evidence of viral proteins in the tumor cells, but not in some of the adjacent vasculature and not in the normal brain adjacent to the tumors. when you use multiple monoclonal antibodies, and then we did every other thing we could think of in situ hybridization, PCR, sequencing, and we kept seeing this virus presence in tumors, but not normal brain. And when it really hit home that this could be important was when Cecilia started saying that her patients were having improved survival with an antiviral drug. And then Dwayne Mitchell and John Sampson at Duke University became interested in this, confirmed our findings, and then basically John Sampson said, look, I don't care if this virus causes brain tumors. I could care less. I just want to find a target for immunotherapy. And the body has an amazing ability to respond to a viral infection. And the work that the Duke group has done suggests that there may be a loss of this immune response that they can reactivate if they vaccinate the patients against some of these CMV proteins. And they had a paper published in Nature last year that showed dendritic cell vaccine against one of the CMV proteins, PP65, in a prospective controlled study. You know, the median survival, at least when this was published, was still around 40-something months for those glioblastoma patients who got the vaccine to CMV. So both of these papers suggest there's something going on that may benefit patients. And my best concept of what may be going on is similar to what has been reported in the way Kaposi sarcoma works, which is you have a virus that's latent most of the lifetime, but under certain circumstances, the virus can get reactivated. In the case of Kaposi's, with immunosuppression in the setting of AIDS, the virus starts producing multiple viral proteins, but it doesn't produce virus, so it's called an abortive lytic infection. These proteins, in the case of Kaposi's, happen to drive all these things like VEGF and anti-apoptotic pathways and multiple cytokines that just shove these spindle cells into a cancer-like state. So if this is the case for CMV, we started to wonder, okay, what viral genes are being produced and could they be participating in these hallmarks of cancer similar to Cecilia? We first noticed if we grow tumor cells in culture, these are cells I had in San Francisco, and stained them for the viral immediate early 1 protein, we would see in cancer stem cell neurosphere assays, we would see viral expression shown green here in the same cells that were Nestin positive, which led us to think that there's some relationship, like Cecilia independently showed, of these viral genes and proteins with the stem cells in the glioblastoma. And that made sense because animal studies that were done in 2002 in mice show that if you infect a mouse with mouse CMV, which has a blue reporter gene, and you reactivate the virus in the mouse by actually slicing the brain and throwing it in culture, the virus reactivates, but not all over the brain, only in specific discrete little clusters in the subependymal zone, which are which are Nestin positive and BRDU positive. In other words, these are glial precursor cells where the virus lives and becomes reactivated. So we came up with the hypothesis that if CMV gets reactivated in a neural stem cell that has a first hit, then maybe it will be able to grow without stopping, not producing virus, but producing all these viral proteins that could drive these hallmarks of cancer and contribute somehow to these cancer stem cell phenomena. This is just an example of a recent study we did in our lab, because we're always trying to make sure we're not crazy. And Demon did a Western blot of 9 glioblastomas and looked with a monoclonal antibody for the PP65 and IA1 protein. They were in all the tumors. And the work we did in San Francisco, what we would typically do is take a tumor from a patient, grow it in neurosphere conditions, then do Western blots. In this case, we thought this IE1 protein I just mentioned might be driving the cancer stem cell phenotype. So we would take tumors right out of patients, grow them in culture, and then knock down this viral gene. Here you can see in the tumor right out of a patient, you have the IE1 protein here. If we knock it down with siRNA, the protein goes away. And we also noticed that all these other proteins, some of them not shown here, that drive the cancer stem cell biology, basically dropped off the Western blot. And the tumor neurosphere cultures essentially stopped growing well, suggesting this viral gene, which drives the whole viral gene expression profile is a master regulator. And we went on to publish a paper last year showing that that's interacting with the critical drivers of cancer stem cell biology. Likewise, if we grow these neurospheres in culture, the virus goes away after a while, at least our ability to detect it. And then if we infect the virus, these neurospheres again, and wait 3 days, as shown here, like Cecilia's data showed independently, we get this robust, massive proliferation of these neurospheres. And we can block that by blocking that viral gene. So the virus is basically like adding gasoline to these neurospheres, and it makes them grow like crazy. Conversely, if you throw this virus on a normal astrocyte, it'll just die. So it doesn't cause a cytopathic viral production, it just causes expression of viral genes that drive the tumor. So like Cecilia, we've independently been looking at a lot of pathways, and we found that the predominant glycoprotein on the cell surface of the virus binds and drives PDGFR alpha, which is a major driver of glioblastoma. We found that the immediate early gene drives all these pathways and inhibits RB and P53. Concurrent with her work on US28, we also found that it drives phosphostat3. And then other colleagues like Amy Heimberger and her colleagues showed that the viral IL-10 is produced in some of these glioblastoma stem cells. So it does fit the picture of a non-productive viral infection that can produce proteins that drive critical hallmarks of cancer pathways. The thing that was fundamentally missing, though, is stepping back from this, and everyone's criticism is like, why don't we see it in TCGA? Why don't we see it when we do certain things? And so I've started a collaboration with Tim Kowalik, who's at the University of Massachusetts, and he's the world's authority really on CMV genome. He's been studying hundreds of samples from children, from the urine, from AIDS patients, from all over the place. He's doing actually very interesting studies looking at fluid from cochlear implant patients who are born deaf because CMV is the #1 cause of congenital brain infection and deafness, and he's been able to amplify up low levels of viral genome and then sequence it. So just recently we sent him the DNA from 6 of our tumors, and he used his technique of amplification and next generation sequencing. And he found in all 6 of these cases, he was able to amplify and sequence nearly the whole genome. And we're in the process of analyzing these 6 glioblastoma CMV sequences. But already we found that there are a lot of alterations that are not seen in most of his CMV samples from urine, from infants, for example. There are areas where there seem to be there are deletions of the virus and maybe even amplifications. We're not sure what this actually means. We're thinking that maybe chunks of the virus might get incorporated into the cellular gene of the tumor and amplified. So it's very early on, but it's very intriguing, and he's kind of blown away by these findings. What he did was just briefly put this into a phylogenetic tree of CMV strains that he's sequenced from hundreds of patients. And what was very interesting, and this is very recent data, is that at least for these 6 glioblastomas, the sequence of all 6 of these tumors was in one tightly defined sort of area compared to urine from infants or some of these other things, strains. This is spinal fluids that he's looked at from patients. So there's a clustering of these CMV genomes that seems to be, at least in the preliminary findings, focused for the GBMs. Maybe this is similar to what Cecilia is finding, that there may be some kind of tumor-associated strain. So we're going to pursue this finding. Lastly, I'll just run through a couple of laboratory studies that we've done that seem to suggest that this persistent infection in tumor cells could be driving important signaling pathways. If we infect glioblastoma cells in the first day or so and look at the viral genes, you get all kind of viral gene expression. But sort of amazingly, these tumor cells can be infected persistently, and you don't even see any viral genes hardly expressed, but you can find some genes that are expressed. And it turns out these genes that we do find expressed after a year or two of culturing and the tumor cells are major drivers of some of the most important cancer signaling pathways. We do RNA-Seq from these tumors, look at the viral expression, and then map it into networks of cellular expression. You see that it's driving the tumors overexpressing certain tumor-related gene products that put it in the microvascular proliferation sort of category of glioblastoma. We're starting to look at the chicken or the egg question that Cecilia talked about. We've gotten some cells that are glial stem cells from colleagues at the Hutch, and they have genetic deletions of some of the signaling pathways involving glioblastoma. And these also, and they have been infected with a virus that has a GFP, so they turn green. So these also stay infected persistently, which is unusual. And we find that after persistent infection of these cells, whereas if you were to infect a normal glial stem cell, you may kill the cell or it won't grow. But after a while, these guys continue to grow robustly, and they start having high levels of expression of some of the major drivers of glioma, cancer stem cell biology, and invasion. And lastly, we've gone right to the source and done some very preliminary work that Dimon, who does a lot of our proteomics work, has elegantly done, which is we've taken tumor biopsy specimens, taken the proteins from those. We've immunoprecipitated viral proteins out of those using antibodies against CMV proteins. So we pull down the CMV proteins from the tumors, and when we immunoprecipitate them, they're bound to cellular proteins and other viral proteins. We've done mass spectrometry to show that we actually can confirm that there are multiple viral proteins in these tumors. And that has been double confirmed by using a SRM type of mass spectrometry where you actually put in a positive control. So we're 100% confident that at least several of these viral proteins are there. But what's even more interesting is when we subtract out the background by doing non-specific antibody and then mass spectrometry, we come up with a bunch of cellular proteins that are bound to the viral proteins. And then in network analysis, we find that these cellular proteins are involved in major pathways that regulate P53, transcriptional regulation, a lot of Warburg effect, metabolism. So the goal of our work in this arena is to go right to the source, pull down viral proteins, see what they're bound to in terms of cellular proteins, and figure out what the microbiome and the interactome is, with the theory that this virus may be an accomplice, may be helping drive the tumor, and that immediately leads to the possibility that if you treat with an antiviral drug and you knock out most of these viral proteins, you may have a benefit by decreasing the aggressiveness of the tumor if you can reverse some of these interactions that are drivers of the hallmarks. Actually, the hallmarks of cancer pathways are all shown in green here, so multiple hallmarks of cancer pathways are being affected. So it's a 15-year work in progress. A lot more work to do, but I'm hoping that by doing this we'll understand the biology, if this virus is actually involved. And it may lead to the understanding of proteins that could lead to further immunotherapy targets and more rational reason for using antiviral drugs. So that concludes all of the talks for today.